All right, so let's finish up with the 5 I's with isolation and inoculating. So isolation technique. Based on the concept that if you take an individual bacterial cell that has been separated from all other cells on a nutrient surface, it will produce a discrete mound of cells, and we call those groups of cells colonies. A colony is a macroscopic cluster of cells that appears on a solid medium arising from the multiplication of a single cell. Therefore, they are all the descendants of that original cell. Therefore, they are all one type of organism. Isolation requires the following things. It requires a medium with a firm surface, such as a nutrient agar plate. It's going to require a petri dish. It's really hard to do isolation techniques in test tubes and other things. Petri dishes are what works well. And it requires you to have what is referred to as an inoculating loop. The isolation technique, you're going to take a mixture of bacterial samples. You're going to get said sample onto your inoculating loop. Hopefully, through this process over here, which you see the streaks going back and forth, that is referred to as a streak plate. Hopefully, while you're doing that, you will separate these cells into discrete one-cell units. You have a single white parent cell there, a single elongated red parent cell there, and you have all the progeny here, all the progeny here, and you wind up with these two colonies that are now visible to the naked eye. Therefore, they are now macroscopic. And here is a surface of a media with the little red colonies growing and then the white colonies growing as well. Sometimes you see they're quite close together, but at other times they're separated, easily recognizable as individual organisms. The method we use is called the streak plate. If you look very closely at this, what this person has done with the inoculating loop is they've gone back and forth over the top portion of this plate. Then they've stopped. They've sterilized that loop again with fire. Then they've come through it going this direction here. And then they will again inoculate by fire and go back this way. Therefore, they've gone several different directions. And then the loop drop is a very similar procedure, but to do a loop drop, what you first have to do is what is referred to as a serial dilution. You take this product, reduce it by a factor of 10, so you've got 1 part of this and 9 parts of distilled water, and then you take this and dilute it by a factor of 10, so you have 1 part of this, 9 parts of distilled water. This is a 1 to 10 dilution. This would be a 1 to 100 dilution. If you did then did this one, you would have a 1 to 1,000 dilution. And if you'll notice, what they are doing with each one of these is they are simply gathering a loop full of this fluid, spreading it all over the surface of the plate, hoping for it to grow. Then another technique is referred to as a spread plate. This is where you take a large drop of culture medium or culture broth, and then you take what is referred to, there's several names for these. There's one called a delta spreader, which has an end, which is a triangle shape, looks like the Greek letter delta. Another one is a bent sort of L structure, which some folks call a hockey stick. We have some in our lab called lazy L spreaders. And you take that, this is a sterile structure, and you use it to sort of paint the surface of that media with that loop full or that quantity of bacteria. All right. Inspection and identification. This is when microbes can be identified through several different ways. You can have the microscopic appearance. What color are the colonies? Are the colonies raised or are they flat? Do they have a slick, shiny appearance, or do they look more fuzzy? Characteristics of the cellular metabolism. Do they metabolize glucose but not sucrose? Do they metabolize sucrose and glucose, but not dextrose? I don't know. Determination of the nutrient requirements. What has to be there for these organisms to grow? And finally, genetic and immunological characteristics. Right. So those are the 5 I's. Now, how big are microbes? The dimensions of macroscopic organisms tend to be given in things like centimeters and meters. The dimensions in microbes, however, range from millimeters, which we are all familiar with, to micrometers, which a few of you may be familiar with, to nanometers, which maybe none of you have ever heard of before. Here's a chart showing you the difference. We are all familiar with a meter. A meter is approximately the same size as a yard. It is a little bit bigger. 1,000 meters is called a kilometer. 1,000,000 meters is called a megameter. And a billion meters is called a gigameter. Now, we will more than likely, well, no, in this class, not more than likely, we will never deal with these 2 units. You may deal with them in a physics class or something like that. The ones we're going to deal with in microbiology primarily are these 3. We have the millimeter, which is 10 to the negative 3rd power, or 0.0001. 0.001. We have the micrometer, which again, some folks will pronounce micrometer because that's the way it's spelled. That is 1/1,000,000th of a meter, or 0.5 zeros and a 1. And then the nanometer, which is 10 to the negative 9th, or 0.8 zeros and a 1. These are the ones that we're going to talk about when we talk about size of microbes. Yeast are generally 3 to 4 micrometers. That little funny looking U right there is the Greek letter mu, and that is the symbol for micrometer. The smallest of the bacteria range from around 200 nanometers to around 750 micrometers. The most viruses measure between 20 nanometers and 400 nanometers, although there are some big viruses that are up to 800 to 1500 nanometers. That's as big as lots of different cells. Again, I will not ask you to remember any of these particular numbers. What I will expect you to remember is that generally yeasts are going to be bigger than bacteria. Bacteria are going to be bigger than viruses in general terms. There are the size of things. Things you can see with the eye. You can see the reproductive structures of bread mold. If you've ever seen the gray fuzzy stuff on bread, that's hundreds of thousands of these little structures. A louse is an individual insect that we collectively call lice. If you've ever, been told that anybody had head lice, that's head lice is multiple, head louse is singular. Microscopic views. There's a red blood cell. There's Giardia namibia. It's a protozoa and causes a condition called Giardia, interestingly enough. Saccharomyces cerevisiae, that's the typical yeast. That's baker's yeast there. Escherichia coli, that's a bacteria. And you see you've got all of these things ranging in the nanometer variety, the micrometer field. And then you have the nanometer things, the DNA diameter, a prion, a flagellum on a bacteria, poliovirus, human immunodeficiency virus, mycoplasma bacteria. And then we have these types of very small things that are measured in nanometers, actually measured in less than a single nanometer. Typical microscope, this looks very similar to the one we use in our lab. Yes, that's the one we use is a little updated version of this, but it has all the same parts. It has a dual or a binocular eyepiece. There's a body, there's a revolving nose piece that holds the objective lenses. The arm is the back portion of the frame back there where you carry the device. The objective lenses are again attached to the revolving nose piece. The stage, this one just happens to have a mechanical stage. And by mechanical stage means it has these two adjusting knobs over here that move the stage forward and backwards and left and right. The aperture diaphragm control opens and closes the iris in this structure called the diaphragm, which lets light from the light source come through. The base is what it sits on. That's probably a light adjuster right there, turned all the way one direction, and it turns the light off. Then as you move it the other direction, the light gets brighter and brighter and brighter. You have the coarse and fine focus knobs, and I believe that's all the parts. I named all the parts. Magnification occurs in two phases. You have the real image, which is formed by the objective, and you have the virtual image, which is formed when the image is projected up through the microscope's body to the plane of the eyepiece. And the ocular lens then forms a second image. Total magnification is derived by taking the power of the objective lens, multiplying it by the power of the ocular, which is the eyepiece, and that gives you total magnification. It is a general rule that the eyepiece almost always has a factor of 10. I worked with some microscopes over at UALR a few years ago. Well, that's no more than a few years ago. A bunch of years ago that actually had an ocular magnification of 13. That's the only ones of those I've ever really seen. It's usually 10. The low power objective on every microscope I've ever worked with was a 10x objective. The high dry objective, again, almost always is going to be 40. Again, although those weird ones I had over at UALR that had the objective power of 13 also had a high dry power of 43 for some reason. So 40 times 10, that's real easy math. 43 times 13, not so easy math. The microscopes we have in our lab for microbiology contain a 100x oil immersion objective. That is an objective that magnifies the item 100 times, but it also requires the use of an immersion oil, which we will talk about in lab a little bit later. And then you take the power of the objective, and if you multiply 100 times 10, you get a total magnification of 1,000. The pathway of light, again, I will never ask you to draw this cutaway version of a microscope. What I want you to see in this structure is that a microscope is a very complex piece of equipment. I cringe every year when I see students taking a microscope and literally dragging it from one side of the table to the other, to the point where as they're dragging it, the microscope is literally bouncing like this. You have an eye, you have a light, and in most cases you think the light is right down here under this objective. It's actually not. The light is back here. The light is actually shining and hits a mirror. That mirror then bounces the image up through the objective, through another set of lenses, where it hits off another mirror here and another mirror here, and then reflects into the eyepiece and the eyepiece lens. So look at all of these different things that could be jostled out of position by that microscope being handled roughly. Resolution, also known as the resolving power, is the capacity of a system to distinguish two objects sitting adjacent to each other or 2 points, one from another. In other words, you see two distinct objects, not one sort of fuzzy overlaid object. The resolving power of the human eye is to about 0.2 millimeters. That's people with good eyesight. As your eyesight goes down, that 0.2 millimeters starts to get even bigger and bigger. So you would think, okay, it's going 0.2 to 0.1, but no, it goes from 0.2 to 0.3 to 0.4, because as you know, some of you may know, as you get older, your ability to see small things diminishes. For instance, I am a coin collector. I like and love collecting coins. In my youth, I had absolutely no problem seeing the dates and what are referred to as mint marks on United States coins. I could tell in an instant what year the coin was made and whether it was minted in the San Francisco Mint, the Denver Mint, or the Philadelphia Mint. I have to use a magnifying glass to do that now, because my eyes simply, my resolving power of my eye is not what it once was. In other words, I can't see things that are 0.2 millimeters. It's more like 0.5 or point. They have to be at least 0.5 or 0.6 before I can diminish what they are. Low resolution. You see, you're seeing the items here, but you get a little bit higher resolution, and you see with greater detail what these organisms look like. How does oil immersion work? Oil immersion works by preventing light from leaving the eyepiece. Here is a large blow up of what an objective lens looks like. The light coming through here, your sample is laying right here on the slide. To see that effectively, you have to get a lot of light to pass through that image or through that specimen. And what happens as light passes through the glass, many of those light rays will want to bend. So you see how these two have hit here and they've bounced back down. This one's come off and it's gone off at a weird angle. And so you've got 1, 2, 3, 4, 5 beams of light represented, but only 2 of them are going through the image and making its way to your eyes. So 40% of the light is doing its job. However, by the use of immersion oil, that's what's represented by this little brown stuff here. Immersion oil, it just so happens, has the same refractive index as glass. The refractive index is how much is that light ray bent as it's passing from mediums of different refractive index. As it goes from light to glass, from glass back into air, from air to glass, then from glass back to air, how much of that light gets bent. But by putting this drop of oil here, we now prevent this from happening. So instead of 40% of the light making it through, going up the tube into the objective, you now have 100% of the light is going to make its way into your eye. The importance of resolution. Here, if you have a microscope that does not have a high resolving power, what you see here is two bacteria, but they're sort of overlapping each other. But with higher resolution, you now see that there are 2 distinct and individual cells. For the larger eukaryotic cell, with the what you see in the microscope is you see this, but what there is in reality is this. You see the little extra colors, and the higher you can get on the resolution power of your microscope, the closer you're going to get to seeing that. Contrast. The degree of bending that light undergoes as it passes from one media to another is referred to as its refractive index. I mentioned that a moment ago. The higher index, the sharper the contrast will be registered. Too much light can reduce the contrast and burn out the image. That's why as you increase your magnification, you actually need to decrease the magnitude of your light or tighten up that iris diaphragm. Control the amount of light going into the condenser. There are also special lenses, such as phase contrast lenses, that help you see things by increasing the contrast. You also can add certain dyes to make those things more visible. Some different types of microscopy, the things, and I will tell you which ones of these we do and which ones we don't. The most widely used type of microscopy is referred to as bright field microscopy. That's what you're going to do with a microscope where you've got your specimen sitting on top of a light. It forms its image when light is transmitted through the specimen. The specimen will absorb some of the light. The rest of the light is transmitted directly into the ocular. Bright field microscopy can be used for both live, unstained and preserved, and stained material. So anything small enough that has the capability of light passing through it can be viewed through bright field microscopy. Dark field microscopy, we will not be doing this. It requires a special condenser that our microscopes do not have. But you do need to know some things about dark field microscopy. These things block the light from entering the objective lens. It will let this peripheral light around the edges, excuse me, and that will come through. The resulting image is particularly striking. Brightly illuminated specimens will be surrounded by a black field. So here's my bacteria on a dark field microscope. Whoops. This is an effective way to visualize living cells. For those of you who are in the lab class, because I'm going to load this for both lab and lectures, I'm almost positive there is a question, you know, which one of these types of microscopy is best used for viewing living cells? Dark field microscopy. Phase contrast microscopes have been constructed to take advantage of the fact that cell structures differ in density. These type of microscopes contains a device that transforms these subtle changes into differences in light intensity. The amount of internal detail visible by this method is greater than either with bright field or dark field, and it's most powerful when observing, or most useful when observing those intracellular structures. So all of those things like this little thing, whatever it is, and this little thing, and this thing over here, all of those things are much more easily seen and studied using phase contrast microscopy. Fluorescence microscopy uses special stains and dyes that will fluoresce under black light. Again, we do not do this particular type of microscopy on this campus because it's too expensive. But it is, again, uses special stains and dyes that will actually fluoresce. Confocal microscopy is another one of these expensive things that most of you will probably never see. Uses a laser beam to show different depths in the specimen, and this delivers a very sharp image that focuses on just a single plane. Transmission electron microscopes, or TEMs. This is the method for seeing things that are exceedingly small or magnifying some things to a much greater extent. For instance, your light microscope may magnify things 1,000 times, whereas your transmission electron microscope may magnify it 35 or 40,000 times. A scanning electron microscope is another type of electron microscope, provides some of the most dramatic and realistic images in existence. Again, very expensive piece of equipment. I have never laid my eyes on a scanning electron microscope. I have seen and worked with a transmission electron microscope. But again, this one has the ability, most of these are connected to a computer, and you can add these colors later You can go in and tell the computer everything that looks like this structure right here, color it yellow, and it will then color all of those things yellow. Then you tell the computer everything that looks like this or this, color them blue, and everything gets colored blue. These colors are added afterwards. All right, I'm going to stop there, give my throat a break, and we will wrap this chapter up in a couple of minutes looking at preparing specimens for the microscope.